Good afternoon, everybody. My name is Gijs Selten. I'm a postdoc at the Utrecht University. And if you were here in the previous session, I'm actually going to introduce the dataset that Petra has used some machine learning upon. And we've been doing SynCom inoculations to study root microbiome assembly. So why exactly is this relevant? Well, in the soil there are many microbes, and some of these can have beneficial effects for the plant, helping them with plant growth, with nutrient uptake, making them more resistant against certain stresses, just to name a few. And so if we want to make use of these beneficial interactions to develop sustainable agricultural applications, we need to have an understanding, how do these microbes interact with each other, with the plant, and how does the root microbiome assemble itself? And we're studying that using synthetic communities, or SynComs. And I'm pretty sure not everybody knows what these are. When you're doing SynCom experiments, what you start with is that you try to isolate as many microbes from the roots as possible when you grow the plant into natural soil. So eventually you'll have a vast collection of microbial cultures, and these you can then recultivate again, mix them together into a synthetic community or a SynCom, which you can then add to the plant. And we're using complex SynComs because very usually you just take between 3 or 10 or 20, but we've actually used SynComs with more than 100 bacterial strains. And the idea is that, of course, this complexity of these synthetic communities is becoming more representative of the natural root microbiome. And another reason why this is important, that if, for example, you're studying how one bacteria colonizes the roots, you might find a certain function that's important for it because it can metabolize these root exudates, for example. However, in different microbiome contexts, this bacterial function might not appear to be so important. For example, if there are other bacteria there that can produce antibiotics against this particular strain. In another context where there would be a bacteria that could neutralize these compounds, this function would again appear in your data as being important for colonizing the roots. And since we want to know how the root microbiome as a whole assembles, we need to investigate the full context of this, and that's why we use complex synthetic communities. So the experiment that we've been doing is called the super SynCom experiment, where we had from 3 different host species, Arabidopsis lotus and barley, also together with maize at the start, wanted to study the root microbiome assembly of these strains, and we created vast rhizobacterial culture collections when they were grown in natural soil. You can see here that we assembled, or we collected almost 1,000 unique bacterial strains that span 25 families and 5 phyla. And here at the bottom, you can see the number of strains that we had for the Arabidopsis lotus and barley maize SynCom from left to right. Also, functionally, these culture collections were quite diverse. In total, we found more than 5 million genes, 5 million bacterial genes in this culture collection that could be collated into more than 8,000 unique KOs. Just more than half of the genes had a KO annotation. You see here in the plot the overlap between the 3 culture collections, and also with every added isolate, how many unique KOs you would add to your to the culture collection, with SSC meaning all 3 together, 3 culture collections. And we also did an all versus all gene alignment using OrthoFinder that led to more than 100,000 clusters of orthologous sequences, or orthogroups. So with the super SynCom experiment, we started with these 3 culture collections from Arabidopsis, barley, maize, and lotus. We cultivated all of these strains and mixed them into 3 large host-specific SynComs, 1 for each host. Then altogether, we also created the super SynCom, and then we inoculated these 3 onto every plant again, in every combination, both under high and low nutrient conditions. And in this moment, we only focused on barley, by the way, because of practical reasons. After 3 weeks, we would harvest the roots, extract DNA, do shotgun metagenomic sequencing, and map these reads back to the bacterial genomes to investigate the microbial composition. Can we quantify all of these bacterial strains from the culture collections? And since we also had information on the genetics, what kind of genes all of these bacteria have, we actually extrapolated this and could also investigate the functional composition. Which bacterial functions can we find back around the roots? And with that, we wanted to answer mainly 2 questions, at least for the talk for today. Can we find certain principles of root community assembly, for which we focus on the KO data set? And secondly, what functions make individual bacterial strains root competent, so being able to colonize the roots, or competitive, being very good at colonizing roots, being in high abundance in the root microbiome. So to start with the first question, we found a variety of interesting principles, but I'm just going to talk about one. Where we first started with doing a PCOA analysis on the data set. You can see here two plots, one for the taxonomic composition on the left, and one for the functional or KO composition. And the colors and the shapes indicate if we're looking at which SynCom inoculation and which host species. And what we already saw here, that there was quite some overlap, both on the genus composition and functional composition. So we tried to quantify that using a per MANOVA analysis. And that tells us for the different things like the inoculum or SynCom type, the host species and nutrient condition, how much these parameters microbiome compositional differences. And what we found really interesting is if you focus on these inoculum R² values, you can see that the one for the taxonomic R² is much bigger than the functional R². So that means that even if you give these three host communities that consist of different genera, different genera assemble around the roots, that in terms of functionality, they are more similar. There are more similar KOs found around the root. So that sort of, we think that this shows that root microbiomes are converging functionally, which might make a lot of sense, but it has not been shown yet in an experimental setup of this scale. For the root competent bacterial functions, what we then wanted to investigate is which functions of the bacteria seem to be enriched in the best root colonizers, the colonizers that have a high abundance in the roots. And we define that by comparing the root community to the input inoculum. So we did a metagenome-wide association study where we tried to statistically associate these orthogroups, these clusters of orthologous sequences, to a high value from root community compared to the input inoculum, because a high value means that since the start, this bacteria really propagated a lot in the root microbiome. And we found a lot of genes that were statistically associated with this, so we also tried to focus on the genes that are located consecutively on the bacterial genome, because these could be indicating biosynthetic gene clusters that, of course, have a much bigger impact on the bacterial phenotype. So the main result from that is shown here, where on the x-axis you can see the best root colonizers. You just have to believe me that these were the best ones from in the data set. And on the y-axis you can see the gene clusters that are statistically associated with competitivity in the root microbiome, so with this high fold change. And you can see here that a lot of these biosynthetic gene clusters can be found into multiple strains, and also multiple of these clusters can be found into one strain. So the most interesting findings here were that many of these biosynthetic gene clusters are related to metabolism. So it seems that if bacterial strains have a diverse metabolism, that they are more competitive in the root microbiome. There are clusters related to motility, so like with flagella, that allows bacteria to swim towards the root, and chemotaxis, and also secretion systems seem to be important. So a sort of secretion systems are used by bacteria to either attenuate or circumvent the plant's immune responses, or to have the sort of elbows out kind of strategy by negatively affecting other resident bacterial strains. And interestingly, you can see also per different synthetic community that it's also different which clusters seem to be important in which context. So to wrap up, I've shown you now here today that we've done these complex SynCom inoculations, and it allows us to study which bacterial functions are important for root microbiome assembly and colonization in a complex microbiome context. I've shown you one principle where it seems these root microbiomes are converging functionally, and that biosynthetic gene clusters that are important for individual bacterial strains to colonize the roots are related to diverse metabolism, motility, and these secretion systems. And with that, I would like to finish. I have to acknowledge the team that has been working on this, with especially Dr. Ronnie de Jong from Utrecht University and Professor Simona Radetuy from Aarhus University. This was funded by the Novo Nordisk Foundation, and if you're interested, we have a preprint online from the first part of the analysis, from the KO analysis. Thank you very much.
Thank you. Thank you. Questions for the speaker? Okay, otherwise I can ask a question.
Sure.
So you do these inoculations and onto it's sterile at the beginning, and then
Yes, it's a gnotobiotic system,
okay.
and the only microbial life in there is the one you add yourself in the SynCom.
Okay. Have you thought? Are you planning to do more where you have some things there, and then you add on top? For example, you add one community, and then you add the second one on top afterwards.
I mean, there are papers that indeed investigate how a resident microbiome can be invaded by a subsequent SynCom inoculation.
Yep.
It's something that we can pursue, of course,
Okay,
but there are
but
also other groups working on that. Yeah,
Right.
I can give you some tips for reading if you want.
All right, no, yeah. All right. So I think with that, well, let's thank the speaker again and give it over to